So good morning. Today we are going to discuss molecular diagnostics. Here, what is the meaning of diagnosis here? Diagnosis. So first of all, the diagnosis is nothing but as a detection here. So detection. So you are taking it is nothing but as a detection. So what we are going to detecting here means that is nothing but as a here we are detecting into the pathogen. So pathogens are we are going to detecting by a process. That process is called as a diagnosis here. So why we are detecting into the pathogens means generally the pathogens are causing in the different types of the diseases. The pathogens may be they are the virus, bacteria, so fungus, or the protozoans, etc. Whatever we are taking means. So all these microorganisms they are causing into different types of the diseases we are. So to detecting into these pathogens, these disease causing pathogens, so we are detecting to estimate so what is the pathogen which is conducting into the what type of the disease which is occurs into the human. So we are going to do so a detection process that is called as a diagnosis here. Generally the diagnosis measures into the a traditional method of the diagnosis we are taking. So diagnosis occurs at the time of so disease symptoms forming. So when we are taking so the diseases we are identifying, the pathogens or the disease we are identifying at the time of the symptoms are forming that particular disease. Symptoms are appears into the patient, that time only we are going to so identifying into the diseases here. So it is not in the earlier stages. Earlier stages we are not going to detecting by the natural traditional methods of the diagnosis we are doing. So after getting of the disease only we are going to detecting or diagnosing into the so disease causing pathogens we are taking. But in this molecular diagnosis, what is expands into the molecular diagnosis means so whenever we are going to diagnosing or when we are going to detecting into the pathogen after the formation of the symptoms, so to control or the cure into these microbes or these pathogens, it is very difficult. So whether we are identified into the early diagnosis of the pathogens or disease causing agents. So what is the major aim of the molecular diagnosis means early diagnosis here. Early diagnosis. The early diagnosis of the disease causing pathogens we are conducting by using of the molecular diagnosis methods we are taking. So first of all, in conventional methods, that means the traditional methods of the diagnosis, what we are doing, so to identifying into the disease causing pathogens, virus, bacteria or whatever it may be, so these disease causing pathogens, how can we are detecting means the diagnosis we are doing by analysis of serum, urine and body fluids here. So generally we are taking and analyzing into the serum and the urine and sometimes in some of the body fluids we are going to analyzing here. Here in the serum urine or the body fluids, they contains the pathogens. Whatever it is a disease that they are causing into the pathogens, those pathogens are presented into these body fluids here. So generally these are conducting the analysis of the serum, analysis of the urine or the body fluid analysis. This method of the diagnosis we are conducting into the laboratory. So we are conducting into the laboratory here. So in this laboratory, the sample of the patient, that is a serum sample, urine samples we are taking. So normal laboratory techniques we are analyzing here. So there we are detecting into the pathogens. Pathogens are we are detecting here. But in these pathogens, when we are going to detecting by normal traditional methods, that time the pathogen concentration here. So concentration of pathogens becomes into the very high. So these are into the very high concentrated here. Why these are into the very high concentrated means? So when we are going to detecting into this serum, urine or the body fluids means whenever the particular person is getting into the symptoms here. Symptoms means the disease characters. When the person is showing into the symptoms, disease symptoms, that time only we are going to analyzing into these samples here from the patient here. So at that time already the pathogens their concentrations becomes into the very high number. The pathogens are presented into the serum or urine or the body fluids we are taking. So that whether we are going to detecting into the early, that means when they becomes into the very low concentrations, it is easy to treat into that particular disease here. So it is easy to treat into that particular disease. So that only we are following into the molecular diagnosis method we are taking. So molecular diagnosis method, generally what type of the diseases we are going to detecting into the molecular diagnosis means infectious disease and genetic disorders. So we are taking infectious diseases and the genetic disorders. So that means heritable disorders or heritable genetic disorders. These two types of the diseases early we are going to detecting. So for this process of the early detection of the disease causing pathogen or the diseases or the genetic disorders, the scientists are working into so many years to early detecting of the diseases here. So for specific and the sensitive detection of the pathogens here. Generally the conventional method of the diagnosis we are taking, it is indirect method. Indirect method here. So it is a indirect method here. So that is into the conventional method or the traditional method of the diagnosis we are taking. It is an indirect method and the laboratory tool method it is. So here we are not detecting into the early, but the scientists are looking for the specific. So specific and sensitive and simple diagnosis here. So these are searching for the number of years they are searching for the specific, sensitive, simple diagnosis. That means it becomes into the direct and the very sensible here. That means very accurately. Here in the indirect method of, that is laboratory method of the diagnosis we are taking, it is not a specific here. Sometimes the result we are getting into the result into the properly, sometimes we are not getting into the result here. So that is not into the specific. Accuracy becomes into the very low here. So that only it is not a specific here. So it is an indirect method. So that only the scientists they are looking for the specific, sensitive and the simple diagnosis method, and they are developed into these molecular diagnosis measures. They are the rDNA technology or DNA technology, PCR method, ELISA method here. So these methods are developed. rDNA technology. So we can write the small r also. rDNA, recombinant DNA technology, PCR method, and the ELISA. Here, so by using of these methods, we are detecting into the pathogens or pathogen causing molecules. So producing molecules, that is disease causing molecules in the earlier stages here. So that only these are into the very specific and the sensitive ones here. So one by one we can be discussed. In the first one is the PCR. So the first one is the ELISA. So ELISA here. So the full form of the ELISA is enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay here. Enzyme linked immunosorbent assay. So what we are going to do in the enzyme linked immunosorbent assay, where we are using into this method of molecular diagnosis, means generally the enzyme linked immunosorbent assay, we are using the detection of the HIV positive patient. So to detecting into the HIV virus, that is a human immunodeficiency virus, to detecting into the HIV, so we are using into the ELISA diagnosis we are doing here. So that means HIV positive patients are earlier detecting into the HIV. So the HIV positive patient we are using into the ELISA. Here what we are going to do into the ELISA means, the ELISA immunosorbent assay we are taking means, so the name itself is explained that it is an enzyme linked here. Enzyme link. Actually, there is one process is there in our body, that is a antigen and antibody. So these are the two components here, antigens and the antibodies here. Antigens nothing but as a pathogen, or generally the foreign particles we are taking, pathogens here. Pathogen molecule. Antibody, with response to the antigen, so our body is producing it the specific protein molecules. So these specific proteins are called as a antibodies with respect to the antigens. Specific to these antigens, antibodies are our body is producing here. So like this, the antigens and the antibodies are generally the interacting to each other. So this is generally called as a antigen antibody reaction. The antigen antibody reaction is occurs in the body specifically. So by using of this principle, we are developed into the ELISA test here. That is a enzyme linked immunosorbent assay method we are developed here. So how this ELISA is working in this process of the diagnosis, what they are doing means, generally we are taking pathogens, already we discussed early stages. So we are taking concentration of the pathogens becomes into the very low. Early stages, the concentration is very low here. So in this very low concentration of the pathogens is not detectable by the conventional or the traditional method of the diagnosis. So there we are using into the ELISA here. So what we are going to do into the ELISA means, generally in this ELISA technique, we are taking the ELISA techniques. Here we are going to divide it. First ELISA specific antigen. So whenever the specific antigen is enters in our body, so automatically antibody is produced here. Antibodies are produced, and specific antigen is enters in our body, automatically the specific antibodies are producing. So that time they are forming into the a complex. It is called as a antibody, or this is called as a antigen antibody complex. Specific antigen is released, and when specific antigen is enters into the body, automatically their proteins, their specific proteins are influencing into the body to produce into the specific antibodies here. They are nothing but as a specific protein molecules here. So like this antigen antibody reaction is occurring into the body. So in this process of the ELISA, what we are doing means, whenever the antigen enters into the body, automatically the body is producing into the antibodies here. Antigen entering, automatically antibodies are producing. So these two are going to reacting with each other here. These two are going to reacting and forming into a complex structure is forming here. So here in this process of the ELISA, we are taking to the ELISA plane here. So antigen antibody complex. So this is the immune process here. It is an immune reaction. So during the diagnosis, what we are going to do in this ELISA means sample containing antigens here. So the sample containing antigen. That means we are taking into the sample from the patient. So that is the suspected patient. So that sample we are going to adding the ELISA plate here. So ELISA plate. So here when we are going to adding into the ELISA plate, here we are going to adding into the sample here. So when we are going to adding, and then we are allow for the immobilization process. What it is the immobilization means? Here whenever we are adding into the sample, it contains the antigen, specific antigens it contains here. So in this antigen is reacted with the antibody, a specific antibody. So these antibodies are we are adding into this antigen sample here. So that is the process is called as a immobilization. Once we are going to immobilized antigen antibody, so immobilization is occurs. That means antigen antibody reaction we are conducted. So this complex we are allowing for linking with the enzyme, their specific enzyme. This antigen antibody complex is linking with the specific enzyme here. The enzyme like that has a peroxidase here. The enzyme like that has a peroxidase enzyme. So like these peroxidase enzymes allowing for the antigen antibody. So these two complexes are linked with the enzymes here. So this complex, now antigen antibody enzyme complex. So this conjugate complex, this conjugate molecule is ready to using. So we are adding into the reagent like 4-chloronaphthol here. Like this reagents are we are adding. So when we are adding into these reagents, the enzyme is going to coloring into this entire complex molecule. So this entire complex molecule is going to coloration here. That means it is going to staining because of these reagents. These reagents are allowed by the enzyme here. So this is generally the process is occurring. So whenever we are taking the coloration, that is the darkness of the complex is directly proportional to the concentration of the antigens here. So that means the more antigens are presented, that means they are producing the more antigen antibody complexes here. So more antigen antibody complexes are producing means the enzyme is giving it the more coloring to this antigen antibody complex. Suppose they are less antigen, the less number of the antigen, the coloration becomes into the lighter in the color. That means concentration of the color, the darkness of the color is directly proportional to the amount of the antigens present into the particular sample. So like this way we are detecting into the amount of the antigens in a sample here. So that is conducting by these 4 steps into the ELISA. First one is a coating, second one is a blocking, detection and readout here. So these are the 4 steps generally we are doing. So first step what we are doing means here this is a ELISA plate. ELISA plate it is. In this ELISA plate we are adding into the some number of antigens here. That means the patient sample we are going to adding here. So in their wells we are adding. So when we are adding into the wells, so we are using into the buffer, coating buffer we are using. That buffer is when we are adding into this coating buffer, these antigens are absorbing into the well surface. The ELISA plate well surface, wells are present. Wells is nothing but a small pits. These pits are absorbing into the antigens here. That means they are immobilized into the wells itself. They are settling into the wells itself. They are stick on into the wells itself here. So this is a process is called as a coating. Once the coating is over, so they becomes into the immobilized. Then we are going to adding into the antibodies. So for the antigen antibody reaction, whatever we are discussing here, the same thing is happening into the. So in this process we are taking. So after the antigens are settled or they are attached into the enzyme link, so that is ELISA plate, that time we are adding into the antibodies here. Then some of the wells in the ELISA plate, so few wells are filled by the antigens. So some of the wells, whatever they becomes into the vacant. So those wells are blocking by the non-related. So these wells are blocked by non-related proteins. So these are blocking. The vacant wells are we are going to blocking by the non-related proteins. Whether we are not blocking means that time, so those wells, that is the vacant wells are so attaches by the enzymatic molecules here. So that only we are blocking into these vacant wells here. This is called as a blocking method. So after the blocking, now antigens are presented. So the specific antigens are present and we are added into the antibodies. Now antigen antibody complex is developing here. This antigen antibody complex is forming on the enzymatic plate. This is the ELISA plate. This is nothing but as a enzymatic plate here. ELISA plate. So already whatever we discussed, whenever the specific antigen is combined with the antibody and we are allowing to the enzyme. So antigen antibody here allowed. So this complex is going to allowing by the enzyme, reacting with the enzyme, the conjugate molecule is forming. Now here the conjugate molecules are forming on the surface of the ELISA plate. So once the conjugate molecule is formed, we are adding into the reagents. That is a coloring reagents we are adding. So automatically wherever it is the antigens are presented, here in the each and every step what we are doing means removing and wash out here. So with the buffer, removal of the buffer, whatever the buffer we are added, that one we are going to removing and wash out later on. So this plate we are going to washing out here. So like this every step whatever we are doing, in this step when we are doing it, the buffer is removing and wash out. Whatever it is the antigen, they are not specific, they are not linked with the antibodies, they are removed out from in this plate. The next step also the same thing. Whatever the free antigens are presented, they are not bind with the antibodies. They are removed from the in this plate here. So after this antigen antibodies, whatever they becomes forming the complex antigen antibody complex molecules are produced. So these only presented into the in this plate. This one also removal of buffer and washout. So once the antigen antibody complex is formed, it is linked with the enzyme. Again we are going to removing into the buffer and the washout. Finally, the enzyme, antigen antibody complex and the reagent material is forming into the coloring or the concentration of the color on the surface of the in this plate here. So based on this coloring, so concentration of the coloring, so we are estimating into the antigens, the amount of the antigens which are presented into the nano range here. So this is a very important to detecting into the nanograms here, nano micrograms, nano so particles, that is a nano, the smaller amount of the antigens also we are going to detect in the earliest stages by using of in this method. This is called as a ELISA, enzyme linked immunosorbent. So like this the steps are conducting, this is the practical approach. What are the things we are doing into the in this process here. So this is into the theoretically what we are going to do, what principle it is internally it is occurring. So that is, so this is into the theoretically we are doing. Antigen antibody first complex is forming, then the enzyme we are going to allowing for the linking of these two. So enzyme linking is occur and the complex is conjugate molecule is forming. This conjugate molecule is adding into the chloronaphthalene. It is going to coloring into the antigen and the antibody complex. More color is forming means more antigens are presented. Less concentrated color is forming means less antigens are. So like this the amount of the antigens are we are going to detecting here. So this is called as a ELISA. So next one is the PCR, polymerized chain reaction here. Okay, polymerized chain reaction. So it is developing the 1983 Corby here. So the scientist is developing in this technique, that is PCR here. So polymerized chain reaction. This polymerized chain reaction, what is the purpose of the polymerized chain reaction means? So it is amplifying, that means the number of copies of genetic material is producing by the PCR method we are taking. So that means what we are going to do in this PCR means, so already we discussed the PCR and the ELISA both are the early detection methods here. Whenever the antigens are very low concentration into the patient or the one person we are taking, means the pathogen genetic material is presented in the very low concentration here. So those genetic materials we are going to amplifying to detecting into the antigens, whether the antigens are presented, whether the antigens are not presented. So like this we are going to detecting by using of the PCR. Here what we are doing means, whatever it is the genetic material is presented, those numbers of the genetic material we are going to amplify. The number of copies are identical copies are we are forming here. So how it is in this PCR technique we are taking means polymerized chain reaction. So three steps are presented. They are the denaturation, renaturation, synthesis here. So denaturation, renaturation is also called as a primer annealing. So this is also called as a primer extension. So other name of the renaturation is a primer annealing, and the synthesis is called as a primer extension here. So like the three steps we are following. So what we are doing into the PCR means whatever it is, the DNA we want to amplify the copies of the DNA. So generally the PCR technique not only into the molecular diagnosis, even though the forensic sciences and the crime scenes, the different purposes we are using into the PCR here. So there a small amount of the DNA material. Suppose we are taking into the one DNA strand. To amplifying into this DNA strand, so first one, this is into the one DNA copy, a selectable DNA material here. So this DNA sample, so we are going to amplifying here. That means we are producing into the more the number here. How we are going to produce means first one is the denaturation process. So first of all, the denaturation we are doing. So denaturation is nothing but as the double stranded DNA is going to unwinding, separating as two single strands here. So first of all, this double strand DNA we are going to separating. Two single strands are formed. How we are going to separating this double strand DNA means this is the step is called as a denaturation. This process is called as a denaturation. Here, whenever the genetic material, suddenly we are going to increasing their temperature for a few or the fraction of seconds, higher temperatures. When the genetic material is exposed to the higher temperature for a fraction of seconds, automatically the two strands are going to divided, denaturated here. These are going to separating. So this is the step is called as a denaturation. So that time each strand itself is acting as a template here for the synthesis of the new copies or the new copy number. So this each strand is acting as a one template here, DNA template. So once the denaturation is occurs, the two strands are produces here. So these two strands are acting as a templates. Next step is the renaturation or the primer annealing. So what we are doing means here the denatured two templates, single strand DNAs are mixing with the DNA fragments and the primer here, DNA primer. Actually the DNA primer, here we are taking the DNA primer. Tag DNA we are taking. We are using into the tag DNA. So what is mean by the tag DNA means it is also called as a tag DNA polymerase 1 here. It is also called as a tag DNA polymerase 1. So what it is mean the tag DNA means thermally stable here, thermus aquaticus. So tag means thermus aquaticus DNA. So thermus aquaticus. So these are the primitive thermo eukaryotes here. So thermotic eukaryotes. So there the DNA polymerase is synthesizing, that is called as a tag DNA polymerase 1. So this tag DNA polymerase 1 we are using for the renaturation and as well as the synthesis here. So what we are going to do, whenever we are going to separate two strands or produces, these two strands are allowing to the tag DNA. The tag DNA is adding the specific portions of these single strand templates here. The specific sequence of the single strand templates here based on into the So their polarity we are taking. Okay, based on their, so their polarities we are taking. The primer is going to adding, tag DNA is going to adding here. So this is the step is called as a primer annealing. So the primer is going and adding into the specific portions of the a single strand DNA. So it is also called as a primer annealing. This is called as a primer annealing here. Once the primer is attaches into the specific portions, so next the number of DNA nucleotides or the DNA fragments, by using of this primer it is going to extend it here. So that is called as a synthesis process here. So next one is a synthesis here. Actually the primer annealing is occurs, it is also called as a renaturation here. So how we are going to do the renaturation? The denaturation we are conducted by the increasing of the temperature suddenly at the 95 degrees centigrade for the few fraction of the seconds. Once the denaturation is occurs, these temperatures are cool downs, very low temperatures here. So immediately we are cooling down into the temperatures. The genetic material temperatures we are going to cooling down into the temperatures. Very low temperatures we are conducting. So few fraction of seconds. Automatically the two templates are, these two separated templates, DNA templates are going to again the renaturated here. That means they are getting into their original nature here. That is called as a renaturation. So that only these are going to adding or annealing with the DNA primers here. DNA polymerase 1 here. So polymerase 1 they are going to add. Once the primer annealing is over, next one is a synthesis process, or it is called as a primer extension. Already primer is added here. This primer is going to extend it here. So this is into the primer first of in this stand, and this is into the primer of in this stand we are taking. This primer is going to extending by continuously adding of nucleotides here. You continuously adding of nucleotides. The primer is going to extend it here. How it is going to extend it means already we know that the tag DNA or tag DNA polymerase, it is a polymerase 1. The polymerase function is adding of the continuously polymerization only, the continuously adding of the nucleotides here. So automatically the polymerase tag DNA is continuously adding their specific the specific nucleotides into in this template stand, and the same way another stand is also continuously adding here. But the extension of the DNA primer, the different distances, the length it becomes into not into the same into the both the stands here, or every time also it becomes into the different distances the extension is occurs. So like this, what it is happened from one single DNA stand to we are developed the two DNA. So two DNA copies are we are done. So like this, the number of copies by repeating of this process we are doing into the number of repetitions, we are producing the more the number of DNA copies are we are going to double stranded DNA copies are going to produce. So this is the technique is called as a polymerized chain reaction here. Okay, polymerized. So like this denaturation, renaturation and the synthesis process or the primer extension. There the primer is going to extending continuously adding into the nucleotides. So like this two stands, their two stands are going to developing as a 4 is going to develop 8. Like this multiplication is continuously occurring. How many number of copies we want, that many number of copies we are going to producing here.